Okay, we're going to get started. I'd like to welcome you all to this week's Broadcast Institute seminar. It's the last seminar in a series that we've had on membrane technologies. I'd also like to extend a special welcome to some high school students from Westdale Secondary School. The STEM Club has done a little field trip to come join us today for this seminar. With that, it gives me great pleasure to introduce our speaker for today, Professor James McGrath. Professor McGrath received his bachelor's degree in mechanical engineering from Arizona State University. He then did a master's degree in mechanical engineering at the Massachusetts Institute of Technology, or MIT. After that, he did his PhD in a joint Harvard-MIT program in biological engineering. And completed his PhD in 1998. He followed that up with a postdoc at Johns Hopkins University, and then in 2001 started his independent position at the University of Rochester in the biomedical engineering department. He currently leads the nanomembrane research group, which looks at ultrathin membranes for a variety of applications. Professor McGrath is a fellow of the American Institute for Medical and Biological Engineering and was recently named Dean's Professor of Arts, Sciences, and Engineering at the University of Rochester. On top of that, he has started two companies, Singapore and Barbario. These are companies that focus on commercialization of his thin membrane technologies, and also their applications in sort of environmental protection, looking at microplastics in the environment. So today he's going to present his work on ultra-thin silicon membranes and how they enable biomedical research technologies. So,
thank you very much. Thank you, Alex. Thank you all for being here. Thank you for the invitation. I owe a thanks to Professor David Latulippe, who's not here because he's homesick. But I'm here because I met David many years ago when the materials I'm going to talk to you about today were first discovered. We had to suddenly get into the membrane science business. I wasn't a membrane scientist. David was a graduate student and a postdoc during those early days, and I got to know him as part of that community. So it's a pleasure to be here. I'm going to give an introduction to and a little bit of a history of the materials that we developed. And then I'm going to walk through a few of the more interesting applications. This is a real mix of folks here. This can be a pretty engineering talk. I talked to a lot of people today who are more on the materials side. And there's also going to be some biological applications. Interrupt me, ask questions. I'm used to that kind of interaction. I taught sophomores for about 20 years at the University of Rochester, so I'm used to. I like interruptions. Lets me know people are awake. So, oh, now we're not going to be able to advance. I have to do. Okay, all right. So the material that's behind everything that in my professional life these days is illustrated in this cartoon that accompanied a seminal paper. What we had discovered was a way of making an ultra-thin porous membrane material out of silicon. Most membrane materials are made out of polymers, and they are, if you look at them in cross-section, very often sponge-like in their cross-section. So that means if you're trying to move a molecule or fluid from one side to the other, it's actually sort of what they call a tortuous path from one end to the other. This thickness means resistance. It slows down the processing of whatever fluid you're trying to filter. And the reason this was such a big deal at the time is we actually made these membranes 15 nanometers thick compared to the conventional 10 microns thick for a traditional filter. So orders of magnitude, many orders of magnitude difference implied that we should see incredible permeability. Our pore sizes are also well-defined, well-controlled. And these are the reasons that this really sort of launched us out of the gate with a new enterprise. The materials are made on a wafer scale. So there's 400 little chips. They are actually membrane chips. The reason these materials haven't taken over the world despite these great properties, which we've confirmed, is because they're very, very tiny. And here that is compared to a simple dime. So we really make chips in the same way that you make a computer chip, but we make membrane chips, freestanding materials on the membrane. This is a typical chip. These are freestanding windows where the porous material that looks like this in ultrastructure is actually suspended. The rest of it's actually a little silicon frame taken from the silicon wafer in which we manufacture it. So we can use the frame to actually house it, mount it into devices. So we are down the road quite a ways. I'll show you that shortly, but we've really we've done an awful lot in the 15 years since this was published. We learned how to really scale the manufacturing. We start making a variety of materials between 50 nanometers and 400 nanometers, what we tend to work with these days. Pores also no longer only in the small scale, we make all the way from 15 nanometers up to 20 microns. These are, in fact, exceptionally permeable. There's a long history of this, that literature that we established, that the materials are exceptionally permeable to diffusing species and pressure-driven flow, but they also have other properties which are really important: optical transparency, electron transparency. They're quiet in a lot of spectroscopic modalities. They're tolerant of chemicals, temperatures, and all these things make them really different than a traditional polymer membrane. So here's a little history. So we started this in 2007, and it actually took us about 7 years to realize materials that had the potential to actually live up to the things we promised in this seminal paper. We had to basically go back in the lab and learn how to make the materials better and better. A thin membrane, as you can imagine, this thin is also frail. And so the problem was we really can only make very tiny membranes. We still make very small membranes compared to conventional membranes, but they are practical now for actually laboratory style separations. And you'll see lots of examples of that now. So it took us a while, and actually the credit goes to Simpor. This is a company we founded at the same time, basically launched the research enterprise. We knew we needed to make progress manufacturing. That really wasn't for graduate students in a laboratory to do. We started a company to do that. So that was happening at the same time. And in about 2014, things really started to come together, primarily by the switch from pure silicon for the manufacture of the material to silicon nitride, which was mechanically robust, chemically very inert. And then once those materials were in hand, we really could incorporate these membrane chips into lots of devices and start really exploring a lot of the properties. So we make a little device, we call it Stepcon. It's a simple little spin cup. You can put in a microfuge, you could spin materials through our membranes and do simple separations. That shows you that the membranes are now stable, and we can do very practical things. We did very advanced things. This is actually filtering a DNA a molecule and placing our membrane upstream of a single nanopore, which is doing single nanopore sequencing of that DNA strand. We have to uncoil it. So our membranes actually uncoiled the DNA so that it actually was read sort of a base at a time. It's actually done with a group at the University of Ottawa. We made a lot of different devices for many, many collaborators. But now we, after all of that learning, we find ourselves in a really happy place, what I like to call the applications era. So this is what I'm going to talk to you about today. We are actually really driving big research projects with this material in the area of diagnostics. So that is the use of our material to catch things out of samples and actually understand what is there, and also tissue chips. So these are tiny little constructs that resemble barriers in the body, like the blood-brain barrier, the gut, the lung, and many others. The retinal barrier is another chip that we're modeling. So this is where we're at, and there's this is my retirement. So we're got a little ways to go before I got to see what I can get done in the time I have left here. So this is really a Rochester enterprise, not only at U of R, University of Rochester, but RIT, Rochester Institute of Technology, two companies. And worldwide collaborations, and to show you the pace of the work that we've empowered finally by getting this all getting our act together after all this long time. We published 80 papers on these types of materials since the beginning of the enterprise, but 31 of those have been since January of 21, not all by my group. So that's actually a sign of success for me. It's coming from others as well. So here's a look at the sizes of things we make relative to the sizes of small scale biology. So proteins, our very first paper, we actually separated IgGs from BSA. This is a virus that's captured on a nanopore. It's zoomed way in an electron microscope. We also do exosomes. You'll see examples of that shortly. Bacteria, these are actually bacteria crawling through these pores. I'll tell you a little bit about that project. It's very cool. And then there's a white cell that's actually being captured on a slit pore membrane that we've done in sort of blood type separations. At the low end, sort of low end in size, our membranes are always less than 100 nanometers thick. They look like nanoporous Swiss cheese. So really high porosities, 15–20% nanoscale, not perfect monodispersed pore sizes. We could talk about that if anybody's interested in what the consequences are, but always a nice cutoff. So there's always a nice cutoff between the size materials that can go through and can't. And then once we get past 100 nanometers or so, we're using photolithography. So this is standard photolithography. We're really good at making membranes with photolithography, but the methods are really not, they've been around for decades. Here we can pattern things absolutely precisely, put pores wherever we want. We can change the shape. And you're seeing that in this example where the pores are actually long slits rather than circles. So the research areas, as I mentioned, are really two. The tissue chip area, this is an example of a neurovascular unit in the brain where we have basically a vascular compartment, a brain compartment where we have cells that are living in the brain, and then we have this interstitial space where this crosstalk between the brain and the blood actually occurs. And we actually set this all up on one of our chips. So we actually have a membrane here dedicated to this interface. We have a membrane over here dedicated to this interface. We have little inserts that we put in to keep them separate and force them to talk to each other through that interstitial space. So it's a very simple device, but really very profound in terms of what you can learn from it in terms of how cells talk to each other. That's for the brain. We make a similar device for the lung, another one for tendon, another one for bone. And there are other systems coming online all the time. In the diagnostics area, I already explained what this picture is all about. This is actually the microplastics work. It's all I'm going to show you on this. We actually take environmental samples, filter them, do a really nice job of just laying out debris, and then using spectroscopic techniques, fluorescence even, dyes, we could figure out which among the debris are plastics. And actually, we're really contributing this technology to the whole hunt for an understanding of just how much microplastic pollution there is in the environment. This is all built on a long foundation that we worked on all those prior years of membrane science, and also microfabrication and nanofabrication. We'll hear a lot of that in this talk. But trust me, we did all that to get to the applications. All right, so tissue chips. So you might have heard of organs on a chip, microphysiological systems, and then I like to use this term tissue chips because I feel like the others are too ambitious. I don't know that we're actually building an organ. I don't know that we're actually building a physiological system, but we are doing of some sort of model on a chip of a tissue. That's definitely what we're accomplishing. So there's actually this little device, which is called the microsim. It stands for microfluidic system enabled by a silicate membrane. Is actually powering the research in 3 large consortia that are all working on tissue chips. On the left-hand, or on your left-hand side, is the neurovascular unit problem that we're modeling. What we're actually modeling is brain damage in response to sepsis. So when somebody is unfortunate to have sepsis, they often, if they're fortunate enough to survive, they often have permanent cognitive impairment. And the mechanisms by which this happened are unknown. And what we need, since we can't actually look at people going through this, we can't look at their brains, we need models of human tissue in a laboratory in order to try to study how this actually happens. So that's what we're doing. We're putting all these cells together, including white cells that come in are thought to be part of the injury. We're putting all this together on a chip. we're studying bone healing in a very similar environment, but a wound healing environment. This is an injury environment that we're modeling. This is a healing environment. We start with the injury and we try to understand how to heal an injured tendon in this other project. Then finally, there's a model of bone infection. I'm going to tell you a little bit more about this later, but this is a very cool project, which is really the leading edge of our work in tissue chips, because they're actually using our devices to hunt for drugs that actually can interfere with the infection of bone by Staphylococcus aureus. The demand for this material and support of these projects really is being driven by this device. And as I say here, we're going to be distributing about 8,000 of these really over the next year to support all these projects. So this is a huge explosion of utility for what we've been working on. Let me try to explain why people would want to use these super thin silicon membranes for cell culture type applications like the tissue chips. So this is actually what you just watched, is a sort of a shrinking of a membrane in an animation from a typical porous filter size down to the thickest example that we currently make for our silicon membranes. What you saw as it played is that the cells on the backside of it actually came into view. They weren't even visible before. And now the communication and even contact, if the pores are big enough, between the cells on either side, which is how barriers in the body work, it's all enabled by the of the material. So that's true. The other thing that's true, which I think a lot of people in this room could explain if I focused on this, or could understand if I focused on this as part of my talk, but it's really not emphasized anymore, is the transport, the small molecule diffusivity across our membrane is so fast, because the membranes are so thin that it's really as if there's no membrane there for all practical purposes. So a cell releasing a factor to communicate with the cell on the other side, that molecule has it essentially takes no real time to talk to get to reach the other one because of the thinness of the membrane. Compared to all the other timescales in this process, transport across the membrane is infinitesimally small, so it can be effectively ignored. So we have a real barrier, but not a barrier to transport. And we rigorously showed that through a lot of fundamental basic science papers on transport in these membranes, and also very recently our blood-brain barrier paper that we published earlier this year. So thinness, a very fast soluble exchange of factors between compartments separated by our membranes, and then imaging. Light goes through our membranes because they're so thin, much like glass. So that's not true for conventionally thick membranes. So here's a track etch membrane, which is typically used for cell culture applications. That's ours, both in SEM. You can appreciate the difference. I hope the pores are roughly the same size, so you could just use the pore as a scale to go back and forth. This is what you get when you try to look at that in a phase contrast microscope. You can't see the cells. You can kind of tell there's something on your surface, but you really can't see the cells. This is what you see on our material. It's like looking at glass. Same thing with fluorescence in both cases. And in confocal, as you see this thing spin, you'll notice that on the bottom, which is our material, there's cells on both sides, one labeled red, one labeled blue. You can't see the membrane. The membrane is actually thinner than an optical wavelength. It's 100 nanometers. You're not going to see it in any reconstruction. I'll show you another pretty profound example of that soon. Meanwhile, the upper one is the track etch membrane, and everybody who tries to do these reconstructions lives with this big black space, which is occupied by the membrane. So there's a lot of reasons for visualization, for imaging these systems, that people would want to work with our membrane. Here's that other example I was talking about. So when two cells come together to form the blood-brain barrier or any other barrier, they cooperate in the formation of a tissue between them, a membrane between them. A basement membrane is what it's often called. And it's made with polymers, extracellular matrix. Collagen is a typical polymer that's found in these materials. And so what you're looking at here is actually confocal cross-section reconstructed where we plated endothelial cells. They form a continuous monolayer on one side of the membrane. And then these cells called pericytes, which are really the matrix builders in cooperation with the endothelial cells, but they're the ones that do the bulk of the work. And they're living underneath sparsely, and they're generating a lot of matrix. And so you can see this beautiful matrix, which is collagen. This is not cells, this is protein. The blue are all cells being formed in between. And again, you don't see the membrane. Using our materials, then we're able to do really good experiments to figure out which cell type is actually contributing to the construction of these. And this is a profound example in the case of a molecule known as fibronectin, where when we look at the ECs, they don't contribute anything, but when we look at the combination, we can see there's an awful lot. So of fibronectin. So the pericytes are really the primary cell type that's contributing to fibronectin to the basement membrane in the blood-brain barrier. We could learn from this kind of study. All right, this is my favorite example of the cool stuff we could do on our membranes because it's a live cell experiment. So what I'm going to show you here is a movie sequence right here that'll play in which white cells interact with an endothelial monolayer on top of our membrane. The membrane has 60 nanometer pores. The white cells are attracted to the other side of the device where we put a chemoattractant that actually looks like a bacteria, because these white cells are professionals at eating bacteria. So you put a little molecule behind there known as FMLP. It smells to these white cells like bacteria, and they basically settle on the endothelium and try to go get it. They can't squeeze through 60 nanometer pores. They're tens of microns in size. So what's going to happen here in our little device is that as they get to the endothelium, they go underneath it and they flatten like pancakes, and they do their damnedest by just walking around underneath the carpet of endothelium. So here you're looking in the background, you're seeing these are the nuclei of the endothelia. Hopefully you can see it. Everything you see in white is actually a rounded up white cell freshly on the top surface of the endothelium. And we play this movie, you're going to watch them transition. They're going to go underneath, they're going to turn dark, and they're just running around in vain underneath the endothelium as a carpet above them, trying to get to the source. I'll show you later, we have the ability to put a micropore or pattern micropores in this nanoporous background, and they will actually find those micropores and jump to the other side where they're trying to get. So this is a model of an immune response on our chip. It's the reason that in that the model of the blood immune response in sepsis, our chips really are very valuable because we can do this kind of study. Not only can we do this, the imaging is so good, isn't it? I'm looking at Jose. It's pretty good imaging. The imaging is so good that we actually are able to, and this actually does take good imaging. We're able to actually program machine learning tools to classify these in a hands-off fashion. So what you're looking at on your right is basically a machine learning algorithm identifying either the leukocytes as either apical, probing, meaning trying to navigate the endothelium, or basal, meaning underneath the endothelium. So we're putting them in one of three states. We can also track them. So that's a state classification. We can count them, track them, get migration parameters as a function of their state. And we can do all of this in a hands-off fashion. So I'll tell you a little bit more about this microstim and why it exists and why it looks the way it does. This is a very engineering part of this talk. So my lab really only operates if we have good collaborators. The stuff we make has to be useful to others. So we've gone through great lengths to make that possible. And back in the day, we were building everything out of PDMS, putting our chips inside, and every collaboration that came along, we said, what do you want? And we made it. And then and we made it very slowly and very painfully, and we shipped it, and they broke, and we shipped more, and they broke. And so you get the idea. I'm trying to convey that this was hard, but fortunately, it was all done by undergrads, so nobody was really hurt, right? So, but it's true. We had, I think, the equivalent of an undergraduate sweatshop operating in my lab to accomplish this. And the problem was every device was different, because we just didn't know what to make. We were trying to follow the collaborators' ideas, and so we had a variety. Very difficult time. And then along one of these projects, sort of the leading project, hit in a sense gold. They wanted 300 of these. Well, we're making a dozen a month if we're lucky. 300 is not a thing. So I finally decided I had to do this differently, and I collaborated with a company called A-Line. They're a microfluidic manufacturer in California that was building microfluidic devices much the way we did in the lab. So I knew there was a hope here that they could automate what we're doing. And sure enough, they did. They were able to basically build these devices to the tune of 100. They shipped the devices, we received them, the yield was great. All the devices were effectively the same. That's important because the further you get into these biological studies, you don't need device variability because biology provides plenty of it. So you need your devices to be the same every single time. Let the biology variability, and if you're working with people who think your devices are variable and they know they have biological variability, they lose interest. So these are hard lessons we learned. But they actually got all the way to using genetic screens to discover the essential genes in these bacteria that were invading bones. And that's why they needed so many devices. But there was a problem. It was scalable in a sense. We were able to make hundreds, but it was only for one of those projects. So this is not still a solution, because if I want to help out the others that want to use with our material, I had to actually still go do it by hand. So we came up with this concept of a modular system. So the modular microsim, in this concept, we mass produce components. We can rapidly change the design of any one component and then mass produce it with the help of A-Line. And then we made it very easy to assemble, ship the kits and the components off to collaborators, even biologists who don't have any microfluidic assembly or microfabrication technique, and they're able to build them in minutes, not days. So this was huge, and this is a reason for the expansion of the use. We're actually putting this in the hands of people who are not engineers and not even really, certainly not in the fabrication sciences. So this is how this works, just really quick. We take our chip, we put it on a little jig. A-Line ships us the components. They're all pressure sensitive adhesives. So steps we're not showing you here is we're actually peeling off these stickers, peeling off these layers that become sticky. And we just press that one on in. We take the bottom part, again, we peel off a top layer. I'm not showing you that. That makes that top piece sticky. We take the top component we had already assembled. We just press it together, and then you can just pull it right out. It really takes minutes. And now you have that little transwell device that I've shown you several times now. And allowing other people to make these rather than forcing undergrads to make it in the lab has changed everything. And so now how do we deal with all the variability that people want to change the device all the time to meet their particular project needs? So the way that works is this: we come up with a concept. Here's a concept for putting bars in a collagen layer so that when the cells contract, they actually don't contract the collagen into a ball. We send the concept to A-Line. They come back with a little design. They make a small production run. We test the production run. It works. I'll explain what this means in a second. But then we basically can go to them for large scale production of components. It's a little misleading. They actually make the components and we can ship and send the components, and the labs actually build their own devices. Here's another example of, this is the example of tendon healing. So this is actually the project. We're looking at wound healing. Again, there's a whole lot of cells in the bottom component. There's these white cells that need to come in as part of the injury response. But if there's too many of them, actually we end up with chronic inflammation, and we end up with scarring. So that in a sense, this project's about trying to learn how to control the whole injury response so we don't get scarring. The problem with the microsim as it was created was that it was very little volume in the bottom compartment, only 10 microliters. And we needed more volume than that. The other problem is we needed anchors to hold the collagen, because as you'll see, that when the cells actually become activated in that bottom compartment, they contract like muscle and they pull the collagen into a ball. And if you don't have anchors holding that collagen, they'll just ball it up, and your experiment basically doesn't work. So we've designed a bottom component with anchors, built it, This is filled with collagen. There's the cells. Here it is after adding a stimulant. The cells contracted. You can see the contraction of the collagen. The anchors worked. You know, now this is their device and it's powering that entire project. There's another component that we can change, and that's the membrane itself. We make 3 primary membranes for the tissue chips. Nanoporous materials, which I explained already. So these are 60 nanometer pores, hundreds of millions of them. This is actually us patterning a micropore in the middle of that nanoporous background, not just one, but thousands or tens of thousands of micropores. That allows the white cells to get across. And then we have these 0.5 micron or pattern pores that we use for the bone project. The neat thing about the micropores when they're put into place is that now the white cell, when it gets under the endothelium, will find one and actually go into the tissue side of the chip. So this is what that material looks like up close. You can see the nanopores are still there. But here's the micropore. And there's, like I said, this is a zoomed in example. The micropores are patterned perfectly over the entire space. Actually, you can see the pattern. There's the micropores. You see them nicely patterned. There's nanopores everywhere. Now, when you're watching this movie, it looks like our prior one, but you'll see this little rippling. You see the rippling? The rippling is and if you look at it very carefully, you'll actually watch the white cells find the holes, get through the micropore holes and let go. And as they fall out of focus, you get this rippling effect. And that's how we know the transmigration is actually occurring. So we're changing the system quickly, changing the chip, changing a component. And now we have a new assay for a new set of experiments. This is the bone project. This is, again, the leading edge of all this. We did this primarily because of the demand placed on our materials and our devices and our undergraduates by this project. And what they're studying, there's a network of cracks in long bone that are naturally there. It's part of a natural healthy bone, but during surgery, bone infection, when it does occur, it's not often, but when it does occur, the Staph aureus, this bacteria, can find its way into those cracks. And when it finds its way into those cracks, it doesn't really easily come out. You're not going to get it out with antibiotics. The immune system can't reach it. The cracks are too small. The mystery of all this was, how does this even happen? The Staph aureus are 1 micron big. The cracks, and our colleagues in the medical center at Rochester, we're finding these cracks filled with this bug. How does this happen? The bugs are not big enough to fit in there, but yet we're seeing them squished into these small spaces, colonizing these networks. Well, they took our membranes, fed bugs to one side, made the pores only 0.5 micron. It's too small for the bacteria to get through. And lo and behold, when they look underneath the membrane in our device, there's the bugs underneath, dividing, getting through. So this was basically a tool now, not only proof that they can do this, they can squeeze in these small spaces and show up on the other side. Now it was a tool. Let's stop that in your device. We have a screening tool. So they did this, identified this particular molecule as a leading candidate, leading gene to go after as a druggable target. They knocked it out of the Staph aureus. No longer does it infect these canals in bone. Our device actually made a prediction that worked. So this is the best you can ask for a system. It doesn't have to be a perfect model of bone. It just has to make a prediction that works. And we did it. So now they're actually using our same system for drug screening. So 30,000 compounds, final stages of the drug discovery pipeline. They're actually always doing functional tests on our membranes to find out, is this actually stopping it? We're doing accessories. So we have a little fluidic plug-in. Just plugs in, we snap all this around it. Now you have flow in the top. So your vascular compartment can actually have blood flow in the top. We have another insert that goes in the top. We've got 2 membranes. I've shown you a neurovascular unit, but here's that barrier that we create that we actually plug in that allows you to culture 1 cell over here and 1 cell over here, and they can only talk by actually secreting factors and talking through the bottom. We've checked it out. There's no lateral diffusion across this. And in the pipeline, we're actually integrating these with ELISA plates. This is an array of our little microsims. This is group work coming out of Michigan. This is another example where we're actually building a 3-dimensional vasculature in the bottom compartment as a model of the blood-retinal barrier. So that's the tissue chip work. And it's just it's wonderful. In the past, since putting these slides together months ago, we've picked up another group that's actually starting to model the gut in our system. So it's just expanding, and it's very, very gratifying. So we talk about sensors and diagnostics. I've got two parts to this that I want to get through in my 20 minutes. There's a number of ways we've used our material for diagnostics, or in combination with sensors, or ultimately as the sensors. There's actually quite a lot of literature we've produced on that. The two I want to talk about are cancer detection using vesicles that are floating around in blood and urine and other body fluids, and then this really fun project on point-of-care pathogen detection. The basic idea is this, and I think this is a universal value, or not universal, but I think there's a lot of value here. We make tiny membranes that are super permeable. What is that good for? It's good for taking a dilute species and concentrating it in a small space, as long as you catch that species, and then taking it over to an analytical instrument and looking at it, right? So compared to any other filter, which is normally much, much bigger, we're going to concentrate everything right in a small space. We've had to learn to do that. That's, in a sense, the principle that's being applied in these projects, and all these projects, actually. So here's a discovery that we can catch cool things in our membrane. It goes back to an experiment we did with undiluted plasma. So this is not whole blood, but it's close. We just got rid of all the cells. What you're left with then is plasma. It's non-coagulating, this version of the plasma, and we just ran it over our device, but we ran it over in a tangential flow system, so that instead of building up a cake of rejected material, anything that's rejected and would normally clog the membrane gets swept downstream. If you don't do that, you end up with this. So just take that undiluted plasma and actually run it directly through the membrane. You pile a cake of protein in milliseconds on top of the membrane. It just clogs instantly. So that's useless. But if you run it in tangential fashion, what we found is that we went back and looked at the membrane. The membranes were occupied by these little vesicle looking things, which we later became aware of were exosomes. And it turns out exosomes are diagnostically like all the rage. Not only diagnostically all the rage, they're also therapeutically a big idea. So what they are small vesicles secreted into the bloodstream, into the urine, into every body fluid by all the cells in your body. All the cells in your body are contributing to this massive pool of small vesicles that are floating around in your body. So it's lead that includes, right, a tumor, if it exists. And even if it's small, it's still contributing to this big pool. But obviously it's diluted in this huge background. So you have to go find it out. You know, find out that there's a cancer there, and you also need to find out where did it come from. But all that information is carried on some of these exosomes. Not all of them. Most of them are useless to this diagnostic, but some of them are carrying information about where the tumor is, even if it's small, right? There's a tumor and it's in your liver. And you can because you have a couple markers on it that you can identify. So this is the big idea, liquid biopsy. But the heterogeneity, the huge background, this is all very difficult. Our idea was, based on that result, let's catch these and then somehow display what's on them. Let's file them in pores like we apparently can, because they're actually that our nanofilters sized perfectly to catch them. And then let's light them up. Let's see what markers they're carrying, and can we turn that into a diagnostic. So before we jumped to doing this with vesicles, we've got a lot of bead work that we've done, and we stopped at that point, went backwards, and just started doing what I would consider the necessary sort of pedestrian basic science in order to make sure we weren't, because maybe this is big and I don't want to fool anybody. So we just captured the gold nanoparticles. So this is gold nanoparticles, 30 nanometer gold, 60 nanometer pores. Managed to somehow get captured in this field. We go to 60 nanometer particles into 60 nanometer pores. You can see the particles are in the pores. You can see that they're actually resting in the pores. So it's a huge difference, same number of particles, but a very big difference in the capture. It's even, so what's happening here is the pores are actually slightly tapered, and we think we're actually tucking them in. even though our membranes are thin, they have some thickness, which is roughly the same size as these beads. So we're actually tucking them in to the middle of that really tiny membrane. What's really cool is we developed we did some tilting to sort of help recreate three-dimensional nature of this. And you can actually appreciate, not only are they in there, occasionally they get caught just on the walls. This must be a van der Waal interaction for that one in the middle, that because it doesn't look like it's being held there by anything else. So we have 100 million pores per chip. So this really means in principle, we should be able to capture 100 million things of interest. We took this all the way to the modeling stage. Here's my membrane science hat. Again, the reason I know people like David is because of this work, which we publish in Journal of Membrane Science, because that's the big one. What we've basically done here is simulate this capture using a particle tracing tool in COMSOL, which just happened to come out when we were doing this work. It's wonderful. So we're actually able to simulate the capture. We're able to line up that simulation, calling that our expectation with our actual experiments. What's really cool in these experiments, once you get to really, really high numbers, notice the membrane actually turns gold. You can visibly see that the membrane is gold. You don't even need a microscope or anything. The membrane turns gold. So we think we understand tangential flow based capture. We call it tangential flow for analytical capture. So now how do we do biomarkers? The way we do biomarkers is we label in solution. So we pre-label everything. We actually label the EV with a luminal label. So it goes into the lumen, and once it's in there, it becomes fluorescent. And then we label the outside with a marker, an antibody that is fluorescent. And we get pictures that look like this. So they're called a starry sky. So you get the green, which means those are your EVs. And because our pores are nicely spaced, I think that might be the next animation. I hope so. No, not yet. I'll back up a second then. But our pores are nicely spaced, so we can actually keep these out of the others way in the image, right? So we can separate them all out. The EVs are green, and then the antibodies are red. So wherever we see orange, we're co-localizing. We can quantify this. This is not hard if we get the image right. This many canned packages can do this. We could do this over. This is what they call a digital assay. When you actually go and figure out diagnostically what's going on, it's what they call a digital assay. It's a clever name, but it doesn't have anything to do with electronics. Everything is electronic. It's just the fact that you're actually counting. You can build your statistics one by one, but of course you can do it in an image very quickly, automated fashion. So we can go all the way from femtomolar, which is really high sensitivity, to picomolar before we start seeing a little bit of an issue. What is that issue? That issue finally is that the pores themselves are 100 nanometer, 120 nanometers. So once occupying all of them, now we're actually hitting the resolution of our system. So we looked at this hard. I'll show this in a second. So we actually have a few methods of capture now. So that this is the fluidic, microfluidic system. This is our microsim. What happened when we started building the microsim is students who were on the other part of the lab, the diagnostic stuff, didn't want to build anymore. They just wanted to use the microsim. So they actually started asking me, when can we build B lines? Right? The company's called A-line. They all called it the A-line. And the folks working on this project were like, we want B lines. But one clever enough student came along and just said, well, you know what? If I just put a furniture bumper literally picked up at Michael's, cover that back port, inject into that one port, it goes into the well. I'll show you more examples of this. And I'll just filter everything right into the well and I'll get my picture. And he's absolutely correct. So this is the absolute simplest way to do this experiment. No microfluidics, just a pipette injection. We can also do this with our spin cups, but then you have to take the chip out and mount it, which nobody likes to do. One thing we do with our spin cups, though, is we pre-filter. So we take our 0.5 micron pores, which are all used for the bugs project, right? And we just clean everything so that we don't get any large stuff. So here is unfiltered. This is an entire membrane. It's 2 millimeters long, 700 microns wide. That doesn't look like a big membrane, but that's a lot of stuff we caught on this membrane. This is the entire membrane imaged on a spinning disc focal. And if we don't do the pre-filtering like this, we get large chunks. If we do, we get a little lawn. You can see this being stitched together. So we stitched it all together. And that's a control on the right. So what we've been doing now is still a lot more of this fundamental work to make sure we're not, you just know what we're doing and we're not screwing anything up. We're just counting beads and putting in beads using the injection format, counting them. There's a million beads in a mill. It's rough. Well, that's still way lower than the amount in blood of EVs. These are just beads, and we're just looking at our ability to predict or count what we put in. And then here comes some limitations as we really, this is 100% capacity, so completely loaded membrane. We figured out for a lot of thought that really we're dealing with optical resolution and pixel size, and we're running into these ultimately at the far end. But we actually start getting some error earlier than that, a little bit earlier than that. We can still do orders of magnitude comfortably and count, but eventually we start running into some trouble. I think it has to do with the fact that our pores are not perfectly spaced, not perfectly sized. We start getting error earlier than we should. I don't know, but still, this work is guiding us so that when we go to do this in a clinical way, we're not overloading the membranes. All right, I can tell you the fun story. I've got 12 minutes. This is the point of care diagnostics. This is a story inspired by failure, which I think is very important for everybody to internalize. So this failure goes. So for years, we were going to NAMS conferences with David and looking at all these huge membranes and thinking, wow, why can't we be like them? Why can't we make desalinization plants? And always looking at these large processes and trying to compete. And the peak of this infatuation with that direction for the research was this interaction with a company, I'm just going to say it, Millipore, right? So I blacked out all that because I was afraid there would be somebody in the audience. So I don't think there is. So this is it. Millipore called us up. We want to try your membranes as virus filtration membranes. Oh, that's awesome. Boy, we're going to hit the big time now. So we sent them a bunch of our little spin cups with our nanoporous membranes, and they took a long time. And then they came back finally and they said, well, two things. First of all, they told me that our permeability compared to their permeability was outstanding. And I was like on cloud nine because nobody externally had ever validated this before. It's been me running around like this, telling people this. I don't know, am I crazy? So here's Millipore telling me I'm not crazy. That was really good news. The bad news is it clogs so fast with virus that the news was rapid plugging, this is a quote, rapid plugging with virus load makes this product uncompetitive. You know, this is actually the data. This is, for those in David's group know this is flux decline. That's their material. That's us. They're declining really fast. All right. So I put this on the shelf and I'm thinking to myself, this can't be all bad news. There's something here, but we have the following. We have rapid clogging from a low baseline, very, very low baseline, right? So we go from nothing. I mean, everything goes through really fast to nothing goes through. That sounds like a switch, right? That's a switch. That's a good thing in engineering. If you could make a switch, a sensor could be. And then along comes COVID and all this money saying, who has novel ideas for how to detect viruses? And I'm like, oh my God, that's it. So I was very fortunate to be able to repackage this quickly, send it off to NSF and get it funded and actually test this as an idea. And it does work. Now, nobody's had a COVID test built with our membranes yet. So blame that on powers that be. But here's the rest of the story. So the concept was simply this. I'm going to build a microfluidic system. Early days, I thought I could just do it with capillarity. We could just do it with the pipette. I've already shown you we can inject the pipette. You'll see more examples of that. The idea is this: the membrane will be so permeable. We'll create the most permeable membranes we ever made, but they will be sized to catch viruses. So when I inject water or sample, it doesn't have a virus. We're going to go down the membrane path and out to a waste. But if it has virus, doesn't need much. That's what I learned from Millipore. I don't need much. It's going to clog the membrane, water's going to reroute, it's going to go hit an indicator, boom. I should get an answer right away whether or not there's any virus in my sample. So that's the other example, flow going around that corner and moving the indicator. To make the membranes ridiculously permeable, we use the slit pore geometry because the long geometry gives you throughput and the narrow one gives you size selection. So rather than use circles, let's make lines and really, really crank up the permeability. So that's what that says. Now for specificity, right, we need antibodies and we're going to get to that. And we knew that. But first, beads. Always do beads first. So we're just going to do steric exclusion, occlusion with beads. So here's our microsim. Yep, it's the microsim again. It just, once you make something easy to use, you can't go back. And that's what's been happening with this device. We barely had started making these when they adopted it for this project. Here's fluorescent particles being trapped on the membrane, and you could just watch us paint the lines. One of the amazing things that we learned through this project is flow focuses everything right to the pores. Not to worry about capture everywhere. Even, it turns out, when the affinity comes online later, this is just sterics. These beads are too big to get through the pores. We could just paint the lines. Here's what it looks like in EM. We're just filling those with the beads. Here's what it looks like when you do the injection. There's two views. This is from the side and this is from the top. You're looking at the well fill through this injection. A little disturbance here just because the pressure went up in the back, but as soon as the injection stops, that goes down. Watch this. Really fill the well with the injection. That's from the side. Here's what happens when you have beads. It just spills out the back port. Okay, there's a little subjectivity here. We need to build in really nice sensors, but it's still a pretty obvious response. So we went with this. To build the affinity, we needed to catch them on the pores. We needed to occlude the pores. So we somehow needed to get the antibody, original thought, was on the walls of our membrane. So as the virus went by, it would be trapped to the walls. That turned out was really hard to do. So, but this is the result. This is the concept. So if you have the wrong antibody, it doesn't have an affinity, it goes right through. If you have the right one, it would clog, and so this is the affinity concept. But it's really difficult. And so what we actually ended up doing is pre-labeling with a streptavidin conjugated antibody, and we biotinylated the chips. So this is the most powerful non-covalent bond that nature makes. Streptavidin biotin, much more powerful than any antibody antigen interaction. It turns out we needed it because we later went and calculated the particles are flying through so fast. There's no time for that antigen antibody interaction to take place. We needed biotin avidin of type affinity to get this to work. We didn't know that at the time, but it's the only way we could solve the problem. We knew how to coat our membranes with biotin. I'm not going into that. Oh, actually, I'm going into that detail. We use this molecule called CODE. It's an amphiphilic molecule. It's biotin on one end. It's hydrophobic on the other end. You literally can just dip the membranes in this stuff and your whole surface becomes biotinylated. We're able to use our spinning disc and focal and actually fly through the pores. And this is fluorescence. And you'll see as you go through the pores, maybe Jose will take issue with my claim, but I think we're actually seeing it on the walls as well. So we were able to convince ourselves that we're getting on the walls. It's everywhere, but I told you the flow focusing says it doesn't matter. You can have biotin everywhere. It doesn't matter. The fluid is going to take your sample and make sure it goes through the pores. You don't have to worry about the fact that it also might stick to the other parts of the chip. So this is streptavidin conjugated beads. It works the same way on a biotin chip. This is actually small beads. Actually, this is us zooming in, so you can actually, if you look carefully, you can appreciate that we're outlining these things. We're actually outlining the pores. So we really are catching these beads in the wall, on the walls. This really, this flow focusing really is working. So then we went through and tested it with beads. At first, we couldn't get it to work routinely. We did false positive and false negative type tests. I'm going to skip some of the details here. Suffice it to say, when we had all three windows working, we had too much capacity and we couldn't get the thing to trip. We had made the membranes too permeable. So eventually what Mike said is, I'm just going to eliminate one of the membranes, and he backfilled it with PDMS, took it out of the equation, redid the experiments, and now we're getting 100% sensitivity, 100% specificity, whether we use sterics or affinity. This is all with beads. Here it is with virus. Now, we're not going to do COVID first thing, right? We're afraid of COVID. So we're going to do vaccinia, which is safe, and we could get buckets of it and the buckets of the antibody from a collaborator. So we did that first. It turns out then we ran out of money, so we never got to COVID. But this is actually vaccinia virus triggering our device. So this actually works. Anti-vaccinia coating on the pores, vaccinia virus being injected to the device, we get the trigger. We actually figured out we could do it over a long range of virus concentrations, which are actually consistent with the pandemic levels of COVID in our system. And we could extend this. If we opened up that third membrane, then we could actually do the really, really crowded concentrated solutions, and those would trigger finally at the very high concentration. So we actually, if we use 2 chips, we actually had a really high dynamic range. Like I said, this is actually, our levels of virus here are consistent with infectious, contagious COVID patients, although it's vaccinia. The one other thing we can do that a COVID test never did is decide whether the virus was intact, because we had a sensor that detected both size and affinity, right? The DNA-based PCR tests break down the virus and just see if the virus DNA is there. People get positive PCR tests weeks after they're infectious, right? The antibody test for antibody, that stuff's circulating in your body too, and it's not virus. It's a product that says you were once infected, maybe recently, maybe months ago, right? It's unclear. This is only going to trigger if the virus, or should, if the virus is the right size and has the right affinity. So we took our virus, our vaccinia. Let's see here. We sonicated it, broke it down. And then basically what we showed here is that we get a switch if it's intact. If we sonicate it, we don't get a switch. So you have to use a label for that. But the level of DNA in the vaccinia virus is the same in a PCR test. We did the PCR test. It doesn't care if you sonicate it or didn't sonicate it, but our device is making that distinction. And again, we had 100% sensitivity and specificity. So that's that. This is my last slide, and I'm exactly on the number. So these are the senior students who actually who are responsible for most of the work done in the group. There's also, I mentioned all our collaborators. A handful of the folks here are particularly showed up in some of the slides at RIT and at Michigan. A number of collaborators at U of R and US and internationally. I highlighted David here. He's not part of these projects, but he has been a collaborator to us. And that's it. Thank you.
Time for some questions. Right.
So in many of these assays, non-specific absorption is a challenge. So how did you overcome? Was there a coating that you provided that prevented non-specific absorption of other proteins that were present?
So we have done that. We've actually developed a silane-based peg coating that does a great job of preventing protein from sticking. The key when you make membranes this thick and you want to do a coating is you cannot block the pores with your coating. So it's not the same as a lot of other coatings. You really have to pay attention, but we were able to do that and show that. It's not easy. So I love this amphiphilic molecule approach. It works great for the microporous materials. Literally, we're dip coating it and the pictures come out like that. I don't know. I was skeptical, but it works. I think I don't think that would work at the nanoscale. So at the nanoscale, we've had to go to grafting PEG molecules using silane chemistries in order to keep that from happening. Yeah.
Hi, Jim. Good to see
Hi.
you again.
Hey, I remember you.
I know you. Fantastic work. Absolutely love it. Quick question for you with regards to your virus capture.
Yeah.
So if I give myself a COVID test, I stick the thing in my nose and it comes out kind of mucousy, right?
Yeah.
Full of all these, I'm guessing.
Yeah. Yeah.
How do you overcome that? I can imagine if you tried to stick that in your membrane and just plug it over.
Yeah, so we worried hard about that and hoped there would be follow-on funding to get to that. And the answer, we never did. We thought the answer was going to, well, two types of answers. First of all, we think we can dilute that sample and still we can make very sensitive sensors. So I think one answer is dilution. I don't know that that's going to be a problem until I see that it doesn't work. That's my plan A. Plan B is to digest the mucins and other things in that mixture to get them broken down so they're not a problem. So we strategized and planned all that. Actually, I've finally resubmitted a proposal. None of you can help me with it. It's obviously going to an American funding agency, but if we get it, that will maybe be able to answer. So great question. Yeah.
Very much.
Kind of a follow-up on that. Like currently, when you take a COVID test, it's kind of like a little bit of a swab, and that's kind of it. For these tests, it seems like with the pipette, it looks like there's like a lot of material going in there. So I'm just like wondering, like, what's the size of it?
40 microliters is all we inject.
Oh, really? Oh,
Yeah.
it's just like the,
Yeah, no, I think it's a small test. Yeah.
Virus sensors, the virus samples containing materials like DNA or host proteins, and does that change the destruction?
So I know our virus samples are inadvertently we're using the brew that's coming from the collaborator's lab. It's not a clean sample. It's got plenty of protein in it. Yeah, so it's got aggregated virus, but that's still the result. So yeah, it's a good question. We would attack it. We would have to see the problem and then try to go figure out what it is. Right now, I know that we can do a pretty dirty solution as long as we get that affinity. We got it. Now, pre-filtration I think that's an obvious, but here's a really interesting step. We could take the same membrane that doesn't have any affinity, right, for the virus and pre-filter the sample. Now, only things, nothing but too big enough to clog it sterically is going to hit the membrane. You see, it's the same membrane. I just didn't put the affinity. Now you take that pre-filtered sample and you run it out. So once we start getting into these more complicated situations, that step is going to be essential. We didn't really need it here. We just kind of opened up the pores and it worked. But I think pre-filtering to eliminate a lot of chunky things is going to be necessary. And then other things that might compete with the affinity we set up, I'm not so sure. Streptavidin and biotin is pretty tough to beat. So I don't know. Keep biotin and streptavidin out of our samples because sometimes it shows up in serum and plasma.
A very impressive talk. Thank you.
Thank you.
I'm curious about the business aspect.
Yeah.
You've invented a new platform, and to get get as far as you've gotten quickly, you've had to work with all sorts of other people. How do you keep the value? How do you protect the IP when you have so many partners?
Wow. So there's only a little bit of core IP, to be honest. One thing I will say about IP in general is it's actually at this point been more trouble than it's worth. It's gotten in the way, right, of moving forward because everybody wants to protect everything I mumble or a student thinks or whatever, and it's paralyzing. And so I actually became a bit sour on the whole idea. it's an obligation. I got to do it. It's part of my job. So when I do have a day that I think is patentable, I tell them. Otherwise, I don't speak too much to the tech transfer office. We just plow ahead. And guess what? If you publish, you ruin it for everybody. Everybody can use it, which actually is kind of the way I'm feeling more and more comfortable. We do have a few things, like the nanoporous membrane is patented. That's pretty unique and it's patented. So I feel good. The nitro and the original one, they're patented. So that's good. Some of the other together, I've gotten ahead and submitted them, but at that point, I stopped worrying about it. I just want to, if this is going to have an impact before that retirement dot becomes, I gotta forget it. It's got to go. We got to make stuff. We got to make stuff, do stuff. So it's a little risky, but it's been too hard to get the IP to work. We've still been able to found companies that have value because as long as they have core IP, and then the company starts filing IP left and right. I don't even know what it's on. So that helps, right? Yeah.
I love to work on the white blood cells moving around, getting under.
Yeah.
My question is there any relevance to cancer metastasis?
Yeah.
Are
Yeah.
you working on that?
No, we're not. It's an example of a model to build. It really is. So I've gone in front of audiences that have that interest, and I've said, look, to study metastasis has to go in both directions across the blood vessel. It has to get into the blood vessel, and then it has to come out of the blood vessel. So our system is everybody. Yes, but everybody's busy and everybody has other things that they have to urgently pursue. So we've never had anybody pick that up. And we can't do it. So still looking for that club who's interested.
Seems well suited for
it is.
screening drugs.
Yep. Yeah, it is. And there's no question, particularly when we start throwing. So the reason we're doing that automation is we're trying to scale everything up, including the analysis, right? You got to scale up the analysis. That can't be what we do, which is, track cells crawling around. That's not acceptable. So once we get the analysis hands off and we get arrays of these things going, we really can turn that exact thing into a screen, no doubt. Yeah.
Okay, with that, I think we'll
Okay.
cut it there. Two things. One is I hope that you will stay for a reception. We should have some food outside and some drinks, and so I hope people will stay and have a chance to chat with a speaker and